Should I just go ahead and start?
Yes, definitely. Do you see your presentation on the screen?
I do. Yep. Thanks, Gary, for facilitating, and thanks, Lauren. Aloha, everybody. So my name is Joshua VanDeMark, and I work for the state of Hawaii's Department of Land and Natural Resources for the Natural Area Reserves Program, and I work with the Hawaiʻi Island Plant Extinction Prevention Program. And I'm going to talk about some of the Cyrtandra species we work with on Hawaiʻi Island, and the examples they provide of questions we often have with a lot of different plants we work with. Questions about relationships between individuals and populations and other taxa, where more information would help us make better management decisions, especially as we're trying to build genetically diverse experimental reintroduction populations. And a lot of this is going to be kind of redundant after Hank's presentation and after the conversation that preceded this. But so I think a lot of the questions we have are similar in a lot of ways to what Hank mentioned, and the other island teams also. But I think but I suspect that some of the answers we might find on Hawaiʻi Island could be a little more complicated or different, just because of where our island is and its evolutionary history and how that's influenced patterns of distribution and relatedness of the plants we're trying to protect and restore the landscape. And it's helpful for me to kind of keep that context in mind when we're trying to understand the patterns of variation we see in the plants we're working with. Next slide, please, Gary. So, as Tutu Pele is currently reminding us, our island is young and still building. Next slide, please. So Hawaiʻi Island's only about half the age of our nearest neighbor, Maui. And as early plant colonists were arriving here, they would have been finding themselves in this vast new landscape with an abundance of novel niche space and opportunities for adaptive radiation. Next slide. And even though the island is young, we're barely a tenth the age of our oldest main island neighbor, Kauai, but we were accumulating endemic species at a really rapid rate. We are approaching almost half the number of island endemics as Kauai in that short time frame. Next slide, please. But at the same time, with our large uniform land mass and the relatively gentle gradients in moisture and temperature, combined with these sort of unimaginable populations of native birds and insects, in probably a frenzy of pollination and seed dispersal, there are these massive opportunities on our island for gene flow across, a relatively immense area. Next slide. So many of Hawaiʻi Island species have these huge historical ranges, even if sometimes, as I've heard from other examples, they're based on a relatively small number of collections across that range. And most of our species have even larger possible ranges predicted by habitat models. So I kind of imagine Hawaiʻi Island as this really dynamic place where adaptive radiation was driving rapid speciation, but also these opportunities for gene flow that would have allowed intergression and combination of lineages. And it kind of seems to me that things were probably really still sorting themselves out here. Then next slide, please. And then, amid that, we showed up with all of our hitchhikers, and in the blink of an eye, geologically and evolutionarily speaking, things were totally interrupted. Land clearing, invasive weeds, predators, ungulates, non-native arthropods, these all led to massive extinctions, fragmented habitats, and rifts in populations. and new barriers to gene flow. So the modern day distribution and the recent historical distributions of species we work with is just this fragment of their original story, and a story that was occurring in a really dynamic, what was probably a really dynamic place. Next slide, please. So from these fragments that we have that are scattered across the landscape, we're often left wondering, you know, if the phenotypic variation we see between populations, or the handful of scattered remnant individuals, reflects, you know, possibly meaningful adaptations to local conditions, or if they are just a small cross-section of diversity in morphology that, you know, once occurred throughout expansive landscape-wide population. And so having better tools to assess these questions would be really helpful in helping us decide where and how to mix founder line representation in restoration populations. Next slide, please. So with that in mind, I'm just going to share some stories of Cyrtandra species that we work with that I think kind of exemplify some of those questions. And again, some of these have already kind of come up in the conversation and from Hank's presentation. But the first story that I wanted to share is kind of a winding one that links, I think, at least 5 of the 9 Cyrtandra species that are known from Hawaii Island. And it starts with Cyrtandra kohalai. So this species was first collected and described by Joseph Rock, and was historically known only from Kohala Mountain, described off of, I think, maybe 3 original collections, which were all centered kind of around Pololu Valley and Puupili. And the manual lists this species as possibly extinct because no records were known since the 1930s. But in 2001, Grant Garish collected this specimen from near Hapahapai Stream, which is near Kapa'au, where Red Star is, kind of in the upper center part of the image. He worked with Linda Pratt to identify it as Cyrtandra kohalai, and in 2003, they sent their barium sheet to Daryl Herbst to help in confirming the determination. But unfortunately, Dr. Herbst was kind of moving into retirement, I think, at that time, and wasn't ever able to review the material. So in the meantime, it remained as considered extinct. And but Dr. Garish took Carl Manyaka to see the plants as part of some of his research in native insect host plants. Next slide, please. And then Carl later found another population where the next star just appeared, matching the description of the other plants in the back of Pololu Valley. And then when Carl saw a list published by Matt Gear and Ken Wood and Dave Lawrence of extinct plants a few years ago, or presumed extinct plants, Carl reached out and said, hey, wait, isn't this Cyrtandra kohalai? And he shared location information of where the plants were. And next slide, please. And we went back into Pololu Valley, and sure enough, the plants were still there. And we collected new specimens, worked through the key, and they really do indeed fit the description of Cyrtandra kohalai. We submitted, updated material to Bishop Museum. So this kind of just highlights, first kind of research question of what tools are out there to help us confirm the identity of individuals and identify the taxonomic identity of some of the plants that we find, especially ones that are thought to be extinct, and being able to help compare them to historical collections. And especially as experts in certain groups may become less active or retire, or when there's disagreement on identities, which isn't unheard of. And so, yeah, just being able to kind of compare to historic collections and help in identifying things. I don't want to discredit or undermine the importance of kind of traditional systematics and looking at morphology, because that's what helped rediscover this species, is someone looking closely at what the morphology was. But I think in order to help confirm these, sometimes it's nice to if we had additional tools. Next slide, please. Actually, Gary, if you could just skip to the next slide. I kind of covered that on the last two slides. So kind of related to that, and the story with Cyrtandra kohalai, the manual mentions that Cyrtandra kohalai, Cyrtandra menzesii, and Cyrtandra gaffardii, all endangered species and Hawaii Island endemics, share similarities, and that the relationship between them should be investigated. It also mentions that Cyrtandra kohalai is similar to, I think, Cyrtandra hawaiʻensis on Molokai. And so kind of in addition to kind of confirming the relatedness of plants to their historic collections, and just being able to kind of clarify taxonomy between closely related species and look further into the relatedness between these different groups would be really helpful. Okay, next slide, please. And you can just skip to the next one, Gary. Sorry, I'm kind of covering the questions, getting ahead of myself. So in our research with Cyrtandra kohalai, we came across this collection. This kind of follows along the lines of some of the things that that Warren was just talking about. So this collection from Scottsburg in the 1930s near Koiave Stream at the back of Waipio Valley, he described a new species from a single collection called Cyrtandra cladantha. And I can understand why in the manual this was determined as a potential hybrid between Cyrtandra platyphila and Cyrtandra kohalai. There aren't a lot of other Cyrtandra species in Kohala, and this one was based on a single collection, and so it probably seemed like a bit of an outlier. But next slide, please. But when I saw this specimen, it really stood out to me as being similar to plants that we were finding in a remote part of Puomi Natural Area Reserve near Lahomeni Stream at the back of Waimanu Valley. And these plants, we were thinking have a strong resemblance to another rare species. Next slide, please. Cyrtandra wagneri, which is named after Warren, and was discovered by Steve Perlman in the early 2000s and described by Steve and Dave Lawrence. This is about 45 kilometers away, kind of in central Hamakua. And all of these plants, Cyrtandra wagneri, these new unknown Cyrtandras at the back of Waimanu Valley, and this single collection of Cyrtandra cladantha, all have kind of some key similarities. Next slide, please. And so coloriferous inflorescences are kind of uncommon on Hawaii Island, at least in Cyrtandra, and all three of these have these flowers originating on the off the branches or the trunk. And I kind of lacked the botanical terminology to describe the shape of the sepals, but I think they're shaped like whatever these things are called that are on a Roman gladiator's leather skirt. They taper outwards, or they expand outwards and then taper at the apex. And all three of these have these really similarly shaped sepals. So, this whole relationship here goes kind of re-emphasizes some of the previous points. You can go to the next slide, Gary. Yeah, just again, just confirmation of a tax's identity. You know, as we're finding these plants in Puʻuʻuminar, we're wondering, okay, these really line up well with Cyrtandra wagneri. Are they indeed? Should we be considering these in restoration efforts where we may combine offspring of these founders, or are they separate taxa that we should manage separately? This also brings up kind of, you know, again, a comparison between disjunct remnant populations or individuals. Is there diversity that would be beneficial to incorporate in restoration, or is it better to keep these segregated? And then also just tools for helping to identify hybrids. Was Cyrtandra cladantha truly a hybrid between Cyrtandra kohalai and Cyrtandra platyphila, or is it a distinct taxa? Is it Cyrtandra wagneri? There's just a lot of complex relationships. If it was a hybrid between Cyrtandra kohalai and Cyrtandra platyphila, that has, you know, broad implications for the previous distribution of Cyrtandra kohalai and what that taxon, where it was and what it was doing on the landscape. Next slide, please, Gary. And then Cyrtandra wagneri, it occurs in Laupoʻohoi at Kilau Stream and was at Kaivilahilahi Stream. Both of these sites also have Cyrtandra tintinnabula, which is another endangered species. And we see individuals here in this population with what seems like intermediate characters between these two. So this kind of complicates, you know, Hank's question about hybrids, where you have a common and a rare species. This is two rare species hybridizing, and it's in a really disturbed site. And, you know, it doesn't have a lot of good outlook for, you know, long-term stability of that site and being able to have these species persist there. And so how do we consider reintroducing these species and their potential hybrids at off-site restoration areas with more intact habitat? Which individuals do we target for collections and reintroduction and so forth? Next slide, please. And yeah, I kind of already covered that. And on to the next slide, please, Gary. And so, and then the story kind of continues with this because Cyrtandra tintinnabula, well, previously was only known from sort of the central Hamakua region of the island. But then in the early 2000s, Linda Pratt discovered, Lauren's letting me know I'm kind of going over time here, so I'll try and wrap it up really quick. So anyway, there's just kind of more examples of how these species and fragmented populations kind of span the landscape and how we can learn more about how to incorporate them into restoration populations. The last thing I just wanted to talk about, I guess, and add to the conversation maybe for the rest of the sessions is to talk about sampling and how, you know, we, like Hank and Matt mentioned, we have a sometimes limited amount of time in the field, and sometimes like specific sampling efforts for a research project can be challenging. And so I'd like to have a conversation with the group more about how we can sample from individuals sort of intermittently as we're doing work with the founders already, and store that material in perpetuity so it can be used as new tools are developed and new new methods are developed. We have Cliff Morton runs the DNA bank here on Oahu, and you know, it'd be interesting to hear more about how that could be utilized as these new tools and techniques are developed, and whether or not that's the best strategy for us to continue adding species or samples to that as we go, as opposed to doing specific targeted sampling efforts. So thanks. I'll end it there. Sorry about the overtime.
Thanks, John.